Okay, great. Good morning, everyone. Thank you for coming at this very early hour. Welcome to our Symposium, Imaging Experience-Dependent Plasticity in Humans. Our speakers today are Svenja Brodt, Valeria Della Maggiore, Heidi Johansen-Berg, and Janif Asaf. And we're going to show some data on how we can actually image plasticity in the human brain using diffusion-weighted MRI. The recent years have shown that diffusion-weighted MRI is actually a very promising method to image neural plasticity, both in gray matter, where we can, for example, observe processes maybe like astrocyte activation or swelling, or even synaptic remodeling and growth that occur over short to long timescales of seconds, minutes, hours, or days. And the idea here is that the formation of plasticity actually impedes the diffusion of water molecules in brain tissue, which we then can observe as decreases in mean diffusivity. We can, however, also image the neural plasticity in white matter, and there we think that this might actually relate to processes like myelin remodeling, myelin formation or changes in fiber organization, and that can lead to a stronger directionality of water diffusion, and we can observe that, for example, as increases in fractional anisotropy. And the speakers of our symposium today will actually show data for both of these kinds of plasticity in gray matter, and Svenja and Valeria are going to show data that relate to this, and I think that Heidi and Janif are also going to talk about plasticity in white matter. And the recent years have actually shown that diffusion-weighted MRI is a very promising tool to image experience-dependent plasticity in the human brain, so how our experience shapes the brain. Here's a couple of publications from today's speakers that showed that over weeks of training, you can actually see plasticity in white matter in the adult human brain. You can also see that faster over only days or hours of training. And the other speakers of this symposium have then shown that you cannot only see this plasticity in white matter, but you can see very rapid plasticity over a single learning session in gray matter, in the hippocampus, and more recently also in the neocortex. And with that, without much further ado, I want to introduce our first speaker, Svenja Brodt, who is going to show how the diffusion MRI can actually reveal the rapid formation of memory engrams in the neocortex.
Yeah, hi. Thank you, Monica, for this very nice introduction. Yeah, as she already mentioned, I'm going to show data from a recent study to show how information derived from diffusion-weighted imaging can actually generate additional knowledge about memory formation and specifically consolidation in the human brain. So memory consolidation, the process of stabilizing newly acquired information, has traditionally been proposed to occur in two phases. One phase termed synaptic consolidation, occurring locally at the synapse, and a second phase called systems consolidation, occurring over much longer time frames and entailing a reorganization of the entire neuronal substrate hosting the memory representation. So traditional models of systems consolidation assume two separate storage sites for declarative memory in the human brain, the hippocampus and the neocortex. And freshly encoded memories are supposed to rely on the hippocampus, which can rapidly store detailed information about an episode. During retrieval of these fresh memories, the hippocampus then reinstates activity patterns in the neocortex that were present during previous encoding. In contrast, neocortical memory traces are supposed to be developed much more slowly, and they require frequent hippocampal reactivation, but in the end they can become independent of hippocampal signaling and then host the memory representation in a stable and enduring format. So what we've been looking at over the past years are the dynamics of systems consolidation and specifically the conditions that can speed up this process. And today I want to focus on the neocortex and the establishment of neocortical memory representations. So the first question is, how do we actually define a memory trace or an engram? And for that, we apply criteria that have been established already in the beginning of the 20th century, but which also have had a recent revival. Accordingly, a memory trace or an engram has to be specific to the learning experience. It has to be stable over time, and it also has to be observable on a behavioral level. There's more to this definition, but I will come to that in a second. These first three criteria we can actually investigate quite nicely by means of fMRI, and we've already done so in several studies now, employing different learning materials from object place associations to spatial navigation in the VR, or also wordless learning paradigms. And we always see the same pattern. So when we look at activity over repeated encounters with information, we can see that neocortical and specifically posterior parietal cortex activity increases steadily from the first encounter onwards. This is specific to the learning experience because it does not occur in a control condition that is exactly similar except for the possibility to learn. And furthermore, we see that this increase in posterior parietal and more precisely precuneus activity is stable over offline periods of about 12 or 24 hours. And you can see that because the activity is increased after the first trial after the offline interval compared to the very first trial in the beginning of the experiment. And this is also specific to the learning condition. And finally, we can also show that precuneus activity is related to successful performance in a variety of ways. So, and additionally, with the help of multivoxel pattern analysis, we can also show in an anatomical precuneus ROI that we can decode the category of the presented stimuli during memory encoding, which actually confirms that this activity is not only experience dependent, it is also content specific. So we've shown that there are memory representations in the PPC that are established rapidly and that fulfill the aforementioned three criteria, but how can we actually know that this mnemonic processing we observe is actually reflecting a true neocortical engram and is not only a mere reactivation or reinstatement of previous encoding activity driven by the hippocampus as would be predicted from traditional models of systems consolidation? Well, we would have to show that this memory representation not only exists while a task is executed, but that it persists in an inactive or dormant state. And if we could do that, we would also fulfill the final criterion for a memory engram, which is that it has to be observable also on the structural level. And this is why we turned with this study to diffusion MRI to image changes in neocortical microstructure over learning. And in this study, our participants had to associate items with their respective locations in pairs on a grid in a task that pretty much resembles the game Pairs or Concentration. They did so in several runs, and each run entailed an encoding and a subsequent cued recall phase. And we had 2 task sessions spaced approximately 12 hours apart. And during these task sessions, we acquired fMRI. The diffusion-weighted measurements occurred at 3 time points. One immediately before learning for a baseline, and then a second one 90 minutes after the first task session to assess short-term changes, and the third one after an additional offline interval of 10 hours, so 12 hours after learning, to look at more stable long-term changes. We also had a control condition following the exact same protocol, just omitting the learning task in between. The results from the fMRI data I've shown on one of my first slides, so we can directly move to the diffusion data and to the question whether the mnemonic processing that we observe is accompanied by structural changes. The short answer is yes, it is. when we look at mean diffusivity from baseline to 90 minutes after the first task session, so for the short-term interval, we would see a significant decrease in mean diffusivity in the posterior parietal cortex. And this is specific to the learning experience, as can be seen by this significant interaction with the control condition. We can show the same effect when we move from the smoothed MRI space data that you see here to the unsmoothed native space of the subject. And what we do here is we look at distributions of mean diffusivity values over all gray matter voxels within a specific region of interest, in this case the precuneus. And you can see here for a single subject that the distribution of mean diffusivity difference values from baseline to 90 minutes after learning in the learning condition, depicted in red here, is shifted to the left, indicating a decrease in mean diffusivity, whereas this is not the case for the control condition in gray. And this effect is present also on the group level when we approximate these distributions by their median. So we can say that there's microstructural changes in the PPC, that they occur rapidly, and they are learning-induced, but in principle, this could just be a transient response related to elevated activity during learning. However, this does not seem to be the case here, because this decrease in mean diffusivity is still present after an offline interval of 10 hours. And this is again specific to the learning condition. So we can say that these rapid changes are persistent for at least 12 hours. And finally, we can also show a relation to subsequent behavior, because stronger short-term changes were associated with better memory retention from task session 1 to task session 2 12 hours later. We see this pattern not only in the posterior parietal cortex, but also in other neocortical regions located along the ventral and dorsal visual streams, which makes complete sense given the task that we've used. And this, by the way, also opened up some really interesting questions about the distributed location of memory representations in the brain. However, to sum it up, the study has shown in two domains, functional activity and microstructure, that there are memory representations in the neocortex that are established rapidly, and they are experience-dependent, long-term stable, and relevant for behavior. And our study also points to a very crucial role of the posterior medial cortex, the precuneus, because this is the one region consistently fulfilling all the criteria that we apply. And altogether, I hope that I could convince you that these early neocortical contributions to memory do not necessarily only reflect a mere reinstatement of encoding activity orchestrated by some other region, the hippocampus, but that they can actually reflect true physical engrams. And besides these direct implications of the study, I also want to briefly highlight why I think that memory research in general can benefit from applying diffusion-weighted imaging. So first, and I hope that I could convincingly show this today, if we combine imaging modalities that together provide measures of online as well as offline memory representations, that can teach us a great deal about how memory formation consolidation actually works. And furthermore, if we are now able to assess memory representations not only while a task is executed, we can actually move to more naturalistic learning paradigms, because we're not restricted anymore by participants having to lie in the scanner during learning and recall. And finally, maybe indices derived from diffusion-weighted imaging can also help us shed light on some of the mechanisms that underlie the beneficial effects of certain factors on memory consolidation, like, for example, sleep. And if you're interested in that, you should visit me at my poster tomorrow at 12:45. And with that, I would like to thank you for your attention. I'm looking forward to the upcoming talks and the discussion. And one last thing, Monica is actually establishing her own lab at the moment. She's looking for PhDs and postdocs who want to image plasticity together with her. And of course, I can only highly recommend working with her. And she's looking forward to hearing from you if you're interested. Thank you very much.